Empty that sucker. So today I'm going to try and do a special edition and talk about a concept rather than papers, and I'm going to look at two papers about this concept. And the concept is grid cells. Grid cells are an in vivo cell type in the entorhinal cortex, and they're thought to encode our sense of space, a vertebrate's sense of space. And their discovery was linked to a long-standing investigation into the function of the hippocampus. And their discovery signifies a sort of turning point in our understanding of the way that the brain works, in the sense of for a long time we've been trying to figure out how neurons work together to encode behavior. We've been trying to figure out how neurons work together to create memories, to facilitate learning. And 1894 was when we first started talking about these connections between neurons and the change of connections between neurons. That was really bad. Anyway, the idea that neurons would connect to one another, and that the connections between neurons would be the connections that support learning and behavior, is a very old concept, more than 100 years old. So it wasn't until 1971, that the first report of place cells in the hippocampus was released. John O'Keefe being the first author on that paper. A very interesting paper in the sense of how the data is displayed, and their sort of characterization of the phenomenon that they're seeing in place cells. But a lot of stuff was happening around 1971, around the same time of the discovery of the place cell. That makes it so interesting is that there were a number of concepts in neuroscience that had yet to find a cellular basis in reality. One of those concepts was Hebbian plasticity, and the idea that neurons that fire together will wire together. Kind of a postulate that goes along with Cahal's idea. And again, this is not a new idea, it's an old idea that's looking for basis in reality in the biology of the brain. And Hebbian plasticity found its sort of seat in biology first in Per Anderson's lab in Oslo in Norway, where the first evidence of long-term potentiation showed up in rabbits' hippocampus. Now, interestingly enough, Edward and Maybrit Moser did their PhD with Per Anderson at the same time as Johan Storm and at the same time as Ulla Paulsen. They all shared that lab together, and it's an interesting thing because every one of those scientists has gone on to be successful. Johan Storm is in Oslo, Ulla Paulsen is the head of Cambridge Neuroscience, and Edward and Maybrit got the Nobel Prize in 2014 with John O'Keefe for the discovery of place cells and grid cells. So that's a pretty nice outcome for your 4 PhD students. Pretty successful. But what are these discoveries, the place cells and the grid cells, and why are they so important? Well, first of all, the hippocampus, at the same time that it was the seat of the discovery of LTP and the seeming confirmation of Hebbian plasticity as a principle of brain function, at the same time the hippocampus was also being investigated because of a patient named HM. And HM is this very famous patient, Henry Mollison, who due to severe epilepsy had some of his hippocampus and most of his entorhinal cortex removed. And after this, he was a pretty functional individual that just simply had no short-term memory and could form no long-term memories. His extremely short-term memory was not that good or awful, and his long-term memories never formed. So he might be able to remember you for a little tiny while, but if he turned his back to you, he probably forgot you. So an extraordinary result seeming to indicate that the hippocampus and parahippocampal cortex is intimately involved in declarative memory, intimately involved in long-term memory storage. So this is going on at the same time, again, that we're looking for a site of Hebbian plasticity, and Per Anderson's lab, along with Lomo and Bliss, are able to detail field potential LTP in hippocampal slices, the same structure thought to be involved in memory. And so this is all becoming very exciting very fast in the early 70s, because all of these concepts are starting to find foundational support in biological observations. So we're starting to think we're going to get a grip on memory, and we're going to get a grip on learning. And so it's at this time that Edward and Maybrit decide that they need to go and study in John O'Keefe's lab, specifically to learn how to make recordings from neurons. And John Eccles is the first guy, I believe, who makes a real single unit recording in a behaving animal. But then the technology quickly, you just need a turn signal. Just need to use a turn signal, and then everybody knows where you're going. So they go to John O'Keefe's lab to learn recordings and start their exploration into the hippocampus. And that exploration is quite fruitful. They do some really nice lesion studies. It's their psychological background, I think, that gives Maybrit and Edward a real edge in the early days in terms of experimental design and so they get a couple really awesome papers before a discussion with Menno Witter leads them to put a PhD student on recording in medial entorhinal cortex. And the reason why they go there is because the main cortical input to the hippocampus originates in the superficial layers of medial entorhinal, and that's a pretty odd It's a pretty odd wiring diagram. There are not very many examples of this kind of odd wiring diagram in the brain where a specific layer of the cortex is projecting a giant projection somewhere else. And in layer 2, just to be specific about it, layer 2 and many other cortical regions at this time doesn't seem to do a whole lot besides local computation. And in the entorhinal cortex, layer 2 sends an exiting signal, a huge axonal projection from layer 2 out of the cortex and into the hippocampus. So that is a significant deviation from how most cortical regions are wired. And Menno found this, characterized it, and suggested that the Moser should put some recording electrodes there because that's likely to be an interesting place to record from. And so the first paper that I think is entitled something like the Spatial Organization or Spatial Tuning of Cells in Medial Entorhinal is Mariana Finn's paper. And in that paper, they first sort of mention that they make recordings in the medial entorhinal cortex, and they see spatially tuned cells with multiple fields. And the main finding that they characterize in this paper is that those fields are as robust informationally as place fields in CA1. And that's the first observation of that kind being made outside of the hippocampus. And it's interesting, of course, because computationally they're connected to the hippocampus, so what are these multi-peak fields? So in Mariana's paper, there are figures that show that the grid cells are primarily in the dorsal medial band of medial entorhinal. That's right now that's no longer known to be not true anymore, but from their original data set and this original set of observations, it appeared to be that the most reliable grid cells, the most robust grid cells, were in the dorsalmost layers. It turns out that the reason why that's the case is likely because the experiments were not done in a way that allowed them to see the grid patterns in the deeper layers of medial entorhinal. The dorsal ventral gradient, not the deeper layers, but the more ventral areas of the medial entorhinal cortex. And as you move ventrally, now we know that those grid fields just change in size. And since Mariana was using a very small open field for the animals to navigate in, it was oftentimes very difficult to see the extent of the grid cell pattern. What are you doing? Holy Christ. Because the room was too small. So in that paper, figure 1 shows the topology to hippocampus from MEC and just gives you a general idea of where MEC is projecting in the hippocampus. And then figure 2 immediately shows that there are spatially active cells, I believe, in medial entorhinal. And then figure 3 looks at the medial and ventral bands sort of moving down the dorsal ventral axis of the medial entorhinal cortex and doesn't see grid cells deeper. That's what that figure 3 is showing. It's probably a mistake, obviously, because they just didn't do the experiments as well as they could have, so that they would have seen what they should have seen, which is grid cells have bigger fields and grid cells have bigger fields and oh, look at this. it up. And so figure 4 compares those fields to hippocampal fields, and then figure 5 actually lesions part of the hippocampus and shows that the spatially stable fields in medial entorhinal are independent of the hippocampus. That data is really cool. So the first paper, the first grid cell paper, then basically shows multi-peaked fields in medial entorhinal cortex and shows that those multi-peak fields are as stable informationally as fields in hippocampus, and even cooler, if they lesion parts of the hippocampus, then they still get the spatially specific fields in MEC, which really suggests that they're intrinsically computing this. This is an intrinsic calculation rather than something that they're inheriting, say, from hippocampus, where previously the only spatially robust data was recorded. So it's really wicked bad. But then on the heels of this paper comes a Nature paper by Mariana Finn's husband, Torkel Hafting, as first author, where they take this a step further, mainly because of discussions with Bruce McNaughton, as I understand it, because Bruce McNaughton is the guy who sort of predicted that upstream from place cells should be a grid-like pattern, and that the grid-like pattern would either be something for path integration or something of an error correction code, or anyway, he predicted the possibility that there would be a type of cell in the brain that would fire grid patterns. And so he suggested very quickly, after seeing the data of Mariana, that they needed to record in bigger rooms and they would see the grid pattern. And so that's what they did. And that's why in Marianne's paper they still don't call them grid cells, but they just say that there is a spatial architecture of some kind in medial entorhinal. I'm sorry that I'm butchering Marianne's paper in that sense. The title, it gives it all away, so I could flash the title up here now and then you would see what I mean. They don't call them grids, they don't mention grids, because essentially they didn't have that data yet. They had an inkling that it might be, but given the size of the box that they were using, they could never see enough fields to verify that the pattern they were seeing was equilateral triangles or hexagonal arrangement of equilateral triangles, which they now see when they make the room bigger. So Torkel's paper is just badass because, again, they show that it's a triangular grid in this paper. So they've got a big enough recording area that they can now see it's an equilateral triangular grid tessellating the environment. And then for figure 2, they show that they can record these grid cells on both sides of the brain. So they show a dorsal, medial entorhinal on one side, and then they show a medial, not so dorsal grid cell on the opposite side. The next thing they look at is multiple grid cells and how their fields are related to one another. And this is another shocking observation in my mind. Because grid cells that are near each other in the microcircuit have very similar spacing, identical spacing and field size. But even more striking, the fields are offset to one another such that no grid cells are encoding the exact same parcel of space. And what that means is you only need a few grid cells to know where in a room an animal is, because the fields don't really overlap. And so you can get a great idea of where the animal is in a room with pretty few cells. And since they fire in more than one location, it's very different than a hippocampal place cell map. Even cooler, they show that the grid cells are present even in the dark, which is pretty badass. That gave them more a reason to argue that grid cells were a sense of space, not completely dependent on vision. And the fact that they're not completely dependent on vision suggested that they are more than that. More than landmarks, but maybe a physical metric. And so the final thing they do is they try to look at the grid cell map in early versus late experience, and statistically it seems that the grid pattern is pretty stable from the beginning of an experience. Which again leaves the concept open that they're really a sense of space. Thanks for joining me on my ride. That was a discussion about the first 2 original grid cell papers from Edward and Maybrit Moser's lab in Norway. The first one is by Marianne Finn and describes spatially accurate cells in the dorsal half of medial entorhinal cortex, and goes on to show that those cells' fields are independent of the hippocampus through lesion. The second paper details more of the microstructure of this map in MEC and goes on to show that the maps are present in both sides of the MEC, that the map is present in terms of different grid cells that do not have overlapping fields. It shows that the grid cells are present even when the lights are off, and it shows, at least statistically speaking, that from their recordings, the early and late stages of the grid cell map are statistically equivalent. This was a huge finding back when it was published, and it remains a huge finding. They got the Nobel Prize for this finding in 2014, along with John O'Keefe, for the place cells in the hippocampus. And of course, this sense of space and its utilization in memory is what at least a large portion of our resources in neuroscience are bent on understanding. The hippocampus and the cortex, and how the hippocampus and the cortex coordinate during memory, and coordinate during recall, and coordinate during learning. And so the idea that there is some higher order spatial processing happening in medial entorhinal that gets delivered via the perforant pathway to all of the fields of the hippocampus was an extremely compelling discovery in terms of trying to then computationally understand how these different in vivo cell types are related. And despite the fact that this was almost 20 years ago now, the discovery of the grid cells maybe 10 years ago, maybe I'm wrong about that. The place cells have been around since 1971, and we're still debating about the ways that a network can be organized from a structural perspective in order to produce that functional output. So this was the start. These two papers were the start of a very long list of discoveries detailed by the Mosers in over 25 almost science and nature papers that proceeded to follow these two papers. So it's worth following up, it's worth reading about. They're excellent papers in terms of how they use behavior and manipulations of experience to dissect the way that medial entorhinal cortex cells encode space. Thanks for joining me on my ride. If you liked it, subscribe, and hopefully I'll see you on my next ride. Take care.